Welcome. My name is Ankersmit. I'm representing a company, an Austrian startup company with the name Aposcience. Aposcience was founded in 2008, and we have developed a secretome-based therapy. Disclaimer. I want to allude to a very well-acclaimed paper authored by Philippe Ménacher. Philippe Ménacher is a French cardiac surgeon who was the first who implanted cardiomyocytes in patients who were undergoing bypass surgery, and he was able to show that these patients are doing somewhat better. In 2001, very famous papers by Kocher and by Orlik were published in Nature Medicine, where it was shown that stem cells were augmenting heart function after experimental AMI. This was a pure sensation, and this sensation went on because clinicians then jumped into clinical trials, and you can read, and you most probably are aware, a lot of publications which basically claimed that if you infuse stem cells or inject stem cells into the heart, most of the time they were autologous, you were able to increase the heart function. In 2015, the picture crumbled a bit because in the CURE study by Marianne Göngeschi, it was basically shown that stem cells has no effect. It's safe, but has no effect after AMI. However, and that's great information, which was basically neglected in the beginning of this hype, stem cell hype. There was one scientist from Duke University, Mr. Gnecki, as first author, showed that basically paracrine factors from mesenchymal stem cells is augmenting heart function. And there was already a twist that stem cells have an effect, but basically it's that what the stem cells produce. In the recent years, it's now commonly accepted by the entire advanced therapy medicinal product sphere that it's not the cell that's homing into the damaged tissue, but it's the cell that is producing a secretome, and that secretome is healing. And that was basically the conclusion of this review article published in 2015 by Philippe Ménacher. What is the current status now with the Alliance of Regenerative Medicine? We know that ATMPs, the so-called advanced therapy medicinal product, are available, are undergoing multiple clinical trials in multiple indications. They are produced under GMP, under a specific jurisdiction, and that is a EC regulation which was published in 2007. So what are ATMPs? ATMPs are somatic cell therapy, genetically modified cell therapies, biological therapies, and also tissues, tissue engineered scaffolds or others. So what does the ATMP? What is the function? What is the mode of action? What is the accepted mode of action? Basically, it was accepted by the regulatory body that ATMPs are living cells. They have multiple ingredients which are secreted, and they also induce multiple mode of actions. In conclusion, mode of action, multiple mode of actions, and also the ATMPs are having their effect via the paracrine factors. What has to be taken into account is that a double viral clearance is not possible. And the concept I will going to show you, which was developed in Austria, is that we are presenting here a novel class of medicinal product, and this is a biological. And we call this biological Apoec, and Apoec contains a myriad of exosomes, proteins, and lipids. It's cell-derived from white blood cells, from stressed white blood cells, and the mode of action is like with the ATMPs, multiple mode of actions which were defined. And Apoec, which is an acronym for apoptotic secretome. Apoptotic secretome from white blood cells can be produced via the autologous route, so patient for his own utilization, or allogeneic. But most important in comparison to ATMP, a double viral clearance can be performed. As you can see on this slide, we had two scientific advices, one with the PAI, Paul-Ehrlich Institute, and one with the Austrian regulatory body, and it was concluded that Apoec is a biological medicinal product. And for that we had to attain all the standards which are basically called upon within this European Union directives, which are here seen on this slide. Respectively, we also had to perform a stability program. In this next slide, I want to explain to you what Apoec is. Apoec is the acronym for apoptotic secretome. Transfusion units are worldwide utilized to donate blood. Blood is then basically separated into the red blood fraction or the plasma fraction. One fraction is completely discarded, and that are the buffets. So the white blood cells are discarded in all transfusion units worldwide. So what we then did was we utilized the white blood cells, irradiated these white blood cells, and cultured this medium with the white blood cells, cultured medium, for 24 hours in order to let these cells secrete all that what is good, what is meant to be good. After that, the cellular compartment gets discarded, and at the end it gets lyophilized. Lyophilized freeze-dried powder containing extracellular vesicles, exosomes, lipids, and proteins. In the left lower side of this slide, you see that the analysis of the donors is most important, and you can basically produce Apoec in the autologous fashion or in the allogeneic fashion. And that's what we did in our company. We directly went for the allogeneic product. That means that basically you're pooling the secretome of multiple donors. In our case, it's about 120. Then these cells get a certificate of analysis after lyophilization, and the quantification goes by protein quantification and validated potency assays. Of course, as I said before, you also have to initiate a stability program, and there we were able to see that our lyophilizate is extremely stable under very good clinical conditions. So it means with +4 degrees, this lyophilizate is transferable, remains its stability and its potency. In this slide, I want to show you the path to clinical application of our Apoec investigational drug product. This is a traffic light: function, safety, efficacy. Function. University research always goes for the preclinical research, the identification of mode of action, where you can show in your research papers why is your drug compound functioning. In the second step, it's yellow, it's safety. Safety is an issue of all regulatory affairs. They want to know, the regulatory body wants to know whether your drug compound, which is produced under GMP, is safe. That means you have to perform toxicology experiments, you have to show the regulatory affairs that you have done a good viral clearance, and you have a GMP facility that produces under good clinical manufacturing conditions your investigational product. And then at the end, there where we now are, we have got the efficacy. The efficacy has to be shown in a phase 1, phase 2 trial. And now in 2020, we are starting with our diabetic foot ulcer clinical trial. Preclinical research, phase 1 research, phase 2 research. First issue. Aposcience has the platform technology, and we have patented in 2009. And we have got this patent nationalized within the entire world. In 2011 to 2015, we have shown that our drug compound, intravenous or topically applied, has a very good effect in indications, preclinical indications such as AMI, stroke, spinal cord injury, and wound healing. Wound healing was our first indication because it's the most feasible one. Since 2013, Aposcience has got a contract with a certified manufacturing organization. This is the organization that basically does the production of our Apoec. And in 2017, first clinical trial in a topical application with secretome Apoec. And in 2020, we're starting with a diabetic foot ulcer study. Here we see a nice illustration of a diabetic foot ulcer. Diabetic foot ulcer is one of these burdens of our humanity. 8% of all world humans has diabetes, and from these 8% of all humans living in this world, 2% will get a diabetic foot ulcer that ultimately can lead to foot amputation. So we have chosen this indication because this is the fastest track to a clinical application. Here I just want to show you one depiction what kind of hazard diabetes is and diabetic foot ulcer. We've got here the atlas, the diabetes atlas of 2017. Here we can see the countries with the incidences of diabetes, prevalences of diabetes in our world. Here you can see Mexico, US, China, India, Sudan. These countries, they have the most diabetic patients, and so therefore unmet need in high power countries. Why did we choose diabetic foot ulcer wound healing? Because we have a clue why this drug is functioning. We know that Apoec is inducing neoangiogenesis. It's causing epithelialization, also manipulates the immune system, and Apoec also contains antimicrobial peptides. The clinical trial is designed as follows. 12 patients will be trialed in the first 4 months, where we include 12 patients in the lead-in phase, and then we're going to have our rollout into multiple institutions, multiple countries within the European Union. This is a depiction how our compound looked. This is here Apoec as a lyophilizate, and this is the clinical entity which is examined in our clinical trial now. Just for information, our clinical trial is named MASIAS 2. At the end, I would like to make a summary. We, as Aposcience, have a disruptive platform technology. White blood cells, waste product of transfusion units, is the source of our secretome. We have a strong IP situation portfolio, which is going to have its validity up to 2037. We have a certified manufacturing organization. And most important, our product is an off-the-shelf product with low cost of good. So you don't have to expand somatic cells, you don't have to expand mesenchymal stem cells, you don't have to expand whatever cells you are currently using within the alliance. It's just white blood cells, which are a waste product. Multi-billion-dollar market, as I've said, diabetes and its sequelae has a tremendous opportunity if this therapy is working. Last, smart management. We were able to get a therapy, a secretome therapy, through the agency, and now we are in the approved phase 2 trial. Near-term milestones, of course, the results are upcoming, and we are looking for partners. And with these words, I want to thank the audience for the attention. Thank you very much.